Hello everyone, I hope you're having a great day. So in this video, we're going to be going over the little laboratory here on the cell. So if we were in the classroom, what I would have you do for this one would be to draw your own cell, label all the organelles, and write a short description for each organelle. One other thing I'm going to go through today is the cell membrane because it's very important. We're going to go over the different components of the cell membrane. So I know this is mostly General Bio 1, but some people have had it a long time ago, so I want to go over all this again because understanding the structure of a cell is very important in understanding anatomy and physiology, because a lot of time when we get to the different tissues, different systems, I'll show a cell. And if you look at that cell and look at the components of the organelles inside that cell, you can give you a good idea of the function of that cell and how it plays in that system. Take the red blood cell for an example. It's anucleate, meaning it doesn't have a nucleus. So it can have a biconcave shape and squeeze through small capillaries. Take B cells or plasma cells are filled with rough endoplasmic reticulum. Rough ER is a protein factory, and B cells are secreting antibodies. They secrete so many antibodies they end up killing themselves from overexhaustion in a way. But we'll talk about that once we get to the immune system, just showing you the importance of understanding a cell structure compared to its function. So structure and function is everything. But right now, let's start at the center. So at the center of the cell, we have our nucleus. So I'm drawing this in blue because typically when we look at a stained cell, the nucleus is blue. Inside here, we have all the DNA. I'm just going to draw it as little chromosomes here, but it would be uncondensed, and this would be completely blue throughout. I plan to make another video after this going over mitosis, so we'll talk a little bit more about the chromosomal arrangements during mitosis. So right there's the nucleus. It's a nuclear envelope. This is a double membrane here. I didn't draw the double membrane, but just know that it's a phospholipid bilayer. So we have the DNA in there, or inside the nucleus. There's also the nucleolus, which is more specific for RNA. And then so the DNA is wrapped around proteins, and it's referred to as chromatin. So make sure you're able to label and understand the different parts inside here. Now, right outside the DNA, let me change color here. Let's go to orange. It's usually right attached to the nucleus. It's called the rough endoplasmic reticulum. And it's just this coiled structure out here, and then it wraps around, and then boom, boom, boom. Now I know this isn't a perfect, usually these channels are connected. This isn't going to be a beautiful cell, so just understand that. These are also, I'm going to draw these blue too, blue for a reason. Little spots on it. So all these have little spots all over it. And why are they blue? Because they would stain blue because they are nucleic acids, and they are ribosomes. So there are ribosomes that are floating free in the cytosol. So I know I didn't draw the rest of the cell yet, but there will eventually be free ribosomes floating around, but then there are also ribosomes attached to the rough endoplasmic reticulum. So the ribosome is a protein factory to cells for protein production, or translation is being done. The messenger RNA comes out of the nucleus, goes to a ribosome, and gets made into a protein. Now, some of those proteins are secreted from the cell, so they have to go to the rough endoplasmic reticulum in order to be secreted. So right here in orange is the rough ER, and then the blue is all ribosomes. And the rough ER gets its name because it's studded with ribosomes. Another thing out here, let's switch to green. I guess it could be blue too. No, not this one. We have these other little, they look like, I remember we're doing a cross section where we're cut in half here. Over here would be the smooth ER, just a general representation. So smooth endoplasmic reticulum is really important for calcium storage. We take a muscle cell as an example. Muscle cell has the endoplasmic reticulum in it, or not, the sarcoplasmic reticulum in a muscle cell where all the calcium is stored. The smooth ER is also important for lipid metabolism, and also it can do some detoxification as well. So that's the smooth ER, no ribosomes. Rough ER has ribosomes. Now moving away from that, let's continue with the endomembrane system. So when I say endomembrane system, I'm talking about anything within the cell that has a membrane. So right away from that, we have the next structure here, which is the Golgi apparatus. So the Golgi apparatus, draw a little vesicle here, well, a little membrane structure here. It's like a little layered structure, and there's little vesicles that might be pinching off each one right here. And then right here, let's say this is removed vesicles coming off. So this would be the Golgi apparatus. So what happens is a vesicle is released from the rough endoplasmic reticulum containing proteins or transmembrane proteins and so forth. It goes to the Golgi apparatus. So the Golgi apparatus has a receiving side right here and then a shipping or sending side right here, which then goes to the plasma membrane around the outside. So the Golgi apparatus is thought of the shipping and receiving center of the cell. Now these vesicles that pinch off have different fates they can go on. They could be carrying transmembrane proteins or membrane-bound proteins that need to be put into the plasma membrane. They could be vesicles packed with proteins, neurotransmitters, things that need to be released from the cell, so they're secreting them. Or they could be in the process of making a lysosome. So that's the next structure I want to talk about here, a lysosome. So here, a lysosome, let's draw it since they're like little stomachs in the cell. So here we have a lysosome. So a lysosome is just a simple double membrane organelle. It's like a little stomach. There's a whole bunch of little hydrolytic enzymes in here, and they fuse with food vacuoles that might be coming in, foreign debris and so forth, and they fuse with that and they can digest it and destroy it. So like a little stomach, the enzymes in there work at a very low pH, and that's how they can go through that process. If the cell is initiating apoptosis or autophagocytosis, where it's breaking itself down, then it would have a lot of these lysosomes doing that process. And then continuing with this, we can then go to everyone's favorite. Let's see. Let's go to this color right here. Yeah. We'll draw a couple of them because everyone loves the good old mitochondria. So here's 3 mitochondria. Mitochondria are what make ATP. I know everyone calls them the powerhouse of the cell. That just happens to be because they have this enzyme in them called ATP synthase, which is where ATP is made via a proton pump. Bacteria do not have mitochondria because these mitochondria are actually about the size of a bacteria. So that's why I don't like calling bacteria the powerhouse of the cell, because bacteria don't have a mitochondria. They have ATP synthase, so ATP synthase is the better powerhouse of the cell. So now you have that. Now typically, real close to these mitochondria are these other organelles called peroxisomes. A peroxisome, think about the first part, peroxide. So what they do, sometimes the mitochondria during their electron transport chain, the oxygen, which is the electron acceptor at the end, becomes an oxygen-free radical. That's very dangerous to the cell. So these peroxisomes take that free radical and convert it to hydrogen peroxide via an enzyme called catalase that then converts it to harmless water. Hydrogen peroxide itself isn't safe, but it's safer than a free radical. And then it completes the conversion back to water. There's some other enzymes involved in that too, but that's just a general feature of these peroxisomes. Then what else do we have here? We have, sometimes you can have other vacuoles in here, so just other little storage vacuoles, or sometimes they could just be little, here we'll draw a little vacuole down here, just a little feature down there. It might be just a vesicle that just went through endocytosis. Remember, there are 3 kinds of endocytosis. There's pinocytosis, there's phagocytosis, and then there's receptor-mediated endocytosis. So all those could come in, and then they could be called a vesicle or a vacuole, where little storage happens inside the cell. And then we're almost to the plasma membrane, but a few more things to draw in here. We also have the microtubule organizing center. So let's switch to pink here. I'm running out of colors. So here, there's these little barrel-shaped structures. And sometimes they're a little cross like this. These are called the centrioles, or the group of them is called, with microtubules coming out, is called the centrosome, that whole region right there. So these are microtubule organizing centers. So when I draw microtubules, I'm going to increase my brush size just a little bit. Microtubules are the largest filaments we find in the cell. So let's draw a few random microtubule filaments going around here. These ones become important when we go through mitosis. They're actually the spindle fibers that reach out. They're also the tracks that all the vesicles walk on to get into or out of the cell. It holds the organelles in place, one of the major components of the cytoskeleton. Another filament that we have here, let's go to yellow now, and let's go just a little bit smaller because that's important, because we have these smaller filaments that the size is smaller, just trust me on that. These filaments are called intermediate filaments. Some of these could be made of keratin, some of collagen. They have some various proteins that make them up. Microtubules are made of the protein called tubulin. And then the smallest one, these ones I'm going to draw in red, are the actin filaments. So here would be some actin filaments. Here would be some more actin filaments. These are the smallest ones. These ones are also important for, some cells have little microvilli sticking out, which are little cytoplasmic extensions, and that's actin filaments are pushing that cytoplasm out for that extension, increasing that surface area of that cell. So then we finally get to, I think I got them all. Let me just look real fast. Sometimes those actin filaments are referred to as microfilaments because they are the smallest. And yes, I don't think I missed any. Oh, the fluid inside is also called the cytoplasm. Now we finally get to the outside here where we have the phospholipid bilayer. Now I will draw the bilayer here. Remember, every membrane we drew also consisted of this bilayer. So within this bilayer now I do want to expand this. So this phospholipid membrane is very important. So now I'm going to go on to a new layer here, and let's expand on this bilayer. So let's make sure we're selected, and let's just change color to yellow now and bring our size back up. Okay, so let's look at this bilayer. What all do we have in here? First thing we have in here, I'm not going to draw them all. We have our phospholipids. Remember, our cell membrane is a phospholipid bilayer. There's no bond that holds these together. So the outside is hydrophilic because these heads are polar. The inside is then hydrophobic. So if I were to draw a little phospholipid up here, this would be polar. This would be nonpolar. Now what else do we have in a cell membrane? We have proteins. Let's draw proteins in green. Proteins could be on the outside, so let's say this is intracellular and this is extracellular, or outside. So this is inside the cell, this is outside the cell. So here the green is a protein. This is a peripheral protein. It's on the periphery of the membrane. Now some of these proteins might squeeze into the membrane. So let's erase a little part of the membrane right here. So this protein here actually integrates itself into the membrane. And if we were to draw the phospholipids around this, the phospholipids would be right in here still as well. And then there could be some up here like that, and like that. So that would be an integral protein. Sometimes these integral proteins can span the whole membrane. These are still considered integral proteins. However, they're now considered transmembrane proteins. So here, this would be a protein that spans the entire membrane. These could be channel proteins, these could be enzymes, they could serve a bunch of different functions. But so, we talked about transport, active transport, passive transport, all those things could be done via these proteins. So that's an integral protein and also a transmembrane protein. Also inside this plasma membrane, a very important lipid structure. It has 4 carbon rings and a hydrocarbon tail is cholesterol. So we find cholesterol embedded in here as well in various parts, and that provides rigidity and makes the membrane more stable. So you can't forget about cholesterol being embedded into the membrane. Remember, cholesterol is a lipid, so it's very nonpolar, and it's happy to be in that membrane. Cholesterol is also very important for some hormone production as well. Now, we also have, I'll put an F here, extracellular fluid and intracellular fluid, because that's the correct abbreviations for that. Now we have what are called the glycocalyxes. So these are carbohydrate tails. Some of these glycocalyxes, so let's do our carbohydrates in pink here. Some of these might be attached to a protein. If the carbohydrate is attached to a protein, so these could be a chain of glucoses here, and they could be branched coming out. That's called a glycoprotein. Now, sometimes they could be also attached to a lipid. Guess what these ones are called? A glycolipid. So these ones are really important because they give your cells their fingerprint, pretty much. They let your body know what's self from non-self. So if a foreign cell comes in, they have a different signature of these glycocalyxes on the surface, and your body can recognize them as being foreign. So I think these are most of the components of the cell membrane. Don't forget, you also have the cytoskeletal filaments that are lining the insides in here as well, that are helping hold everything in shape too. But these are the ones I want you to know, because we will talk about a lot of these. We'll talk about how sometimes a protein could be out here. So just an example of how this is really important. We have a protein right here, say transmembrane protein. There's a little binding site right here. This molecule comes in and binds. That binding is called a ligand, sends a signal inside, and it activates a G protein that then sends this other protein, a peripheral protein right down here. It activates it. So it's a G protein, so it goes from GTP to GDP. It's just a little activation pathway. It goes down here to another protein called adenylate cyclase, which then takes that phosphate that's on there and goes and makes cyclic AMP, which then causes calcium release. So lots of stuff going on here. You don't have to know anything I just said there. I was just showing you how all this stuff becomes important later. Understanding the structure of the membrane and being able, and also, I know I was sitting here drawing this. Draw it on your own. See if you can label all the different parts and go through. I know I didn't label names on all this, so make sure you do that as well. But that's all I have for today. So we talked about the membrane here. Then we also talked about just the overall components of the actual cell. So I know it's a very, very dirty drawing representation of it, but it's okay to do dirty drawings. There's still information in that, just as long as you know what you're drawing and you understand the importance of those organelles. Just looking over my notes, I think I hit everything I needed to hit on. So the next video, we're going to be talking about mitosis, and I hope to see you then. So I hope you all have a great day. If you have any questions, definitely let me know. But with that, yeah, bye-bye.